Welcome to the PubMed Daily Review for May 30th, 2026. Today we scan 9,999 new open access papers across 715 journals. In this video, we'll cover 10 trending research topics and 10 selected papers for detailed analysis. Trend 1 of 10, Engineering Materials and Processes. Developing, characterizing, and applying diverse materials and engineering processes for various industrial and environmental challenges. This cluster covers 701 papers. Representative work includes investigation of adaptive interception systems for concentrate and spiral concentrators, pilot-scale remediation, resource utilization, and long-term stabilization of cadmium, lead and arsenic co-contaminated soil, and machine learning-driven evaluation of mechanical and microstructural properties of agro-waste-derived geopolymer concrete. Trend 2 of 10, Infectious Diseases and Microbiology. This trend explores various aspects of infectious diseases, including their control, emergence, characterization, and treatment across different hosts. This cluster covers 876 papers. Representative work includes infection control and prevention awareness among intensive and cardiac care nurses, implications for managers and policymakers, investigating the emergence of the Brazilian porcupinpox poxvirus in wild Brazilian porcupines, Coendu longicaudata, and isolation and characterization of Lactobacillus plantarum LP2301 from bovine milk, acid and bile tolerance, antibacterial activity, and genomic safety evaluation. Trend 3 of 10, Clinical Diagnostics and Interventions. This trend encompasses clinical methods for diagnosing diseases and evaluating therapeutic interventions across various medical specialties. This cluster covers 1475 papers. Representative work includes clinical outcomes of 27-gauge needle-guided flanged intrascleral haptic fixation using the Yamane technique in various indications, chronic pain and fatigue in multiple osteochondroma and all-year disease, a systematic review, and serological biomarkers of JI-associated uveitis, ANA titers, ANA-AC, 30 staining pattern, and pre-foldin 5 antibodies. Trend 4 of 10, Neuroscience and Brain Disorders. This trend explores neurological disorders, their underlying mechanisms, and potential therapeutic interventions. This cluster covers 803 papers. Representative work includes adipocytes and macrophages after stroke, interactions between metabolism and immunity, digital and repetitive transcranial magnetic stimulation interventions for perinatal depression and anxiety, a systematic review, and sigma 1R CD36 axis in myeloid cells contributes to the alleviation of depression-like behaviors. Trend 5 of 10, Public Health and Well-Being. This trend explores diverse factors influencing health outcomes, disease prevention, and overall well-being across various populations and life stages. This cluster covers 1503 papers. Representative work includes correction, physical education and sports teachers' health and well-being, workload, psychological resilience and social support perspective, and relationship between pre-pregnancy body mass index and postpartum depression, and the mediation effects of cesarean delivery, low birth weight, child anomaly, and breastfeeding. A prospective birth cohort in the Japan Environment and Children's Study. Trend 6 of 10, Bio-inspired materials and devices. Developing materials and devices inspired by biological structures and functions for applications in medicine, sensing, and engineering. This cluster covers 692 papers. Representative work includes biomimetic hybrid bioactive glasses gelatin composite rescues alveolar socket healing from diabetic impairment, synthetic biology-driven bio-inspired delivery systems for RNA therapeutics and neural repair, and optimizing DNA origami assembly by reducing off-target interactions. Trend 7 of 10, Molecular and Cellular Disease Mechanisms. Investigating molecular, cellular, and diagnostic aspects of disease processes and pathology across diverse biological systems. This cluster covers 1141 papers. Representative work includes mitochondrial extracellular vesicles between pathology and regeneration. Phosphomanamutase 1 restrains adipose thermogenic programming via inosine signaling, and folic acid attenuates maternal sertraline-induced hepatic injury in rat offspring. Trend 8 of 10, Applied AI and Advanced Technologies. This trend explores the application of artificial intelligence and cutting-edge technologies to address diverse real-world challenges. This cluster covers 737 papers. Representative work includes monolithically integrated photon mapping infrared imager, AI-driven personalization of food systems, from precision nutrition to climate-resilient supply chains, and the evolution of mosquito baiting, from chemical and electronic methods toward AI. Trend 9 of 10, agricultural and animal production, focuses on optimizing animal and crop production through nutrition, genetics, and resource management for improved yield and quality. This cluster covers 899 papers. Representative work includes re-evaluating manganese requirements of broiler chickens under phytase supplementation, asymmetric life history trade-offs shape, sex-biased longevity patterns, and meat quality and muscle fiber comparisons between rapid and slow-growth broilers. Trend 10 of 10, cancer biology and therapeutics. Advances in understanding cancer biology, improving diagnosis, screening, and developing novel therapeutic approaches. This cluster covers 1172 papers. Representative work includes surveillance and surgery for IPMN, an urgent call to challenge the status quo. Molecular advances in juvenile myelomonocytic leukemia and associated RA-sopathy, and enhanced antimicrobial and anti-cancer properties of penicillin G-adapted lacticacea bacillus rhamnosus GG against colorectal cancer cells. Up next, our first paper out of 10. Dual targeting of glutamine metabolism and lysosomal function in eliminating drug-tolerant KRAS mutant cancer cells by Hiroki Furukawa and Shigeki Aoki from Laboratory of Biopharmaceutics, Graduate School of Pharmaceutical Sciences, Chiba University, Chiba, Japan. This paper was selected because it introduces a novel dual targeting strategy that effectively eliminates drug-tolerant persister cells in KRAS mutant cancers, addressing a critical challenge in oncology and significantly advancing. Let's break down its key contributions. KRAS inhibitors are a game-changer in cancer treatment, but a major challenge is that cancer cells can become tolerant to these drugs, leading to relapse. Our study aimed to understand how these drug-tolerant cells survive and adapt. We specifically looked at the metabolic changes that occur, hoping to find new ways to overcome this resistance. We investigated KRAS mutant pancreatic and lung cancer cells in a drug-tolerant state. Using advanced proteomic and metabolomic analyses, we meticulously mapped their metabolic adaptations. Our goal was to identify vulnerabilities that could be exploited to overcome drug resistance. Our findings showed that drug-tolerant cells exhibit significant metabolic reprogramming, particularly involving lysosomes. Crucially, we found that simultaneously blocking glutamine metabolism and lysosome-associated processes selectively killed these resistant cells. This effect was reversed by alpha-ketoglutarate, which appeared to act by supporting redox balance. This study reveals a critical redox vulnerability in drug-tolerant KRAS mutant cancer cells, offering a new therapeutic avenue. Our findings provide a strong rationale for combining KRAS inhibitors with agents that target glutamine metabolism and lysosomal function. This approach could significantly improve outcomes for patients by preventing relapse and enhancing treatment durability. Up next, our second paper out of 10. Mutation-tolerant inhibition of HIV, 1-integrase strand transfer by secondary metabolites from the endophytic fungus Alternaria alternata PO, 4PR2 by Nzalo and Nampumelo P. Mkwanatsi from HIV Pathogenesis Program, School of Medicine, College of Health Science, University of KwaZulu-Natal, Durban, South Africa. This paper was selected because it presents a highly novel discovery of natural product compounds that effectively inhibit HIV, 1-integrase, crucially demonstrating efficacy against drug-resistant strains, which directly addresses a critical and pressing global health challenge. Let's break down its key contributions. HIV-1 continues to be a major global health concern, and the emergence of drug resistance, particularly to integrase inhibitors, poses a significant challenge. This paper explores a promising new avenue, natural compounds from endophytic fungi. Specifically, it investigates whether an extract from Alternaria alternata PO 4PR2 can effectively inhibit drug-resistant HIV-1 and how it achieves this. Researchers designed a comprehensive in vitro study. They created specific INSDI-resistant HIV-1 mutants using site-directed mutagenesis and tested the fungal extract's activity in TZMBL cells. A range of molecular techniques, including luciferase assays and integrase activity assays, were employed to determine antiviral efficacy and pinpoint the mechanism of action. Finally, molecular docking helped identify the key active compounds. The results were quite compelling. The fungal extract demonstrated potent inhibition of all tested INSDI-resistant HIV-1 mutants, with very low IC50 values ranging from 0.02971 to 0.1652 micrograms per milliliter. Crucially, it showed minimal cytotoxicity, with a CC50 of 300 micrograms per milliliter, maintaining over 80% cell viability. Further analysis revealed that the extract inhibited integrase activity by 67%, specifically targeting the strand transfer step, and molecular docking identified coumarin derivatives as the key active compounds. This study offers a significant step forward in the fight against drug-resistant HIV. 1. The identification of mutation-tolerant compounds from a natural source, like Alternaria alternata PO4PR, 2, opens new avenues for drug discovery. While promising, further research is essential to isolate and characterize the specific coumarin derivatives responsible for this activity. Ultimately, these findings lay the groundwork for developing much-needed new therapies to combat INSTI-resistant HIV. 1. Up next, our third paper out of 10, high-fidelity genome and prime editing enabled by the AI-designed OpenCRISPR, one by Hae-Yun Hwang and Dae-Sik Kim from Department of Precision Medicine, Sungkyunkwan University School of Medicine, Suwon, Republic of Korea. This paper was selected because it introduces an AI-designed CRISPR system that significantly advances genome and prime editing by achieving high fidelity without compromising on-target activity or guide RNA design flexibility, addressing a critical limitation in gene. Let's break down its key contributions. CRISPR, Cas9 technology has truly transformed our ability to edit genomes, opening doors to countless research and therapeutic possibilities. However, a persistent challenge has been balancing the system's on-target editing efficiency with its specificity, meaning avoiding unintended edits elsewhere in the genome. Often, improving one comes at the cost of the other. This paper addresses that fundamental limitation, seeking a solution that offers the best of both worlds. Researchers conducted head-to-head comparisons in various human cell lines, including HEK 293T, iPSCs, and MRC5 fibroblasts. They evaluated the novel AI-designed OpenCRISPR, one nuclease against standard Cas9 and high-fidelity Cas9 variants, as well as its prime editor versions against existing prime editors. Key outcomes measured included on-target editing efficiency, the extent of off-target mutations using advanced sequencing methods, and the overall specificity ratio. The system was even tested for delivery using engineered virus-like particles, mimicking a clinically relevant approach. The results were quite striking. OpenCRISPR, 1 demonstrated editing efficiency comparable to standard Cas9 across multiple genomic loci. Crucially, it achieved up to a 553-fold reduction in off-target mutations compared to Cas9, and its off-target indices matched or even surpassed those of current high-fidelity Cas9 variants. Furthermore, when converted into a prime editor, OpenCRISPR, 1 lowered the relative specificity ratio by an impressive 97%, indicating superior precision in more complex editing tasks. These numbers clearly show a significant leap forward in genome editing precision. This research is truly groundbreaking, as OpenCRISPR, 1 represents a significant advancement in genome editing, finally overcoming the critical specificity efficiency trade-off. It showcases the immense potential of generative AI in designing novel proteins with tailored properties, opening a new era for rational protein design. Looking ahead, the next crucial steps involve evaluating OpenCRISPR-1's performance in living organisms and exploring its direct therapeutic applications for various genetic diseases. This technology holds great promise for both research and clinical settings. Up next, our fourth paper out of 10, carbon nanotube hydrogel electrodes for high-fidelity intraoral EEG in wearable neurotechnology by Alexandra Stefania Mihai and Onegaman from Department of Computers, Electronics, and Automation, Faculty of Electrical Engineering and Computer Science, Stefan Selmer, University of Suceava, Romania. This paper was selected because it introduces a novel carbon nanotube hydrogel electrode for intraoral EEG, representing a significant technological advancement that directly addresses the pressing clinical need for discrete, high fidelity. Let's break down its key contributions. Good morning, everyone. Today, we're diving into a fascinating study on intraoral EEG, a groundbreaking technique for monitoring brain activity discretely. This method is particularly important for patients who need continuous, long-term neurophysiological assessment, like newborns or those with neurological conditions. The core challenge lies in developing biocompatible electrode materials that offer low impedance and stable contact for reliable performance. The researchers focused on gel-MA/HEMA hydrogels, which they doped with varying concentrations of carbon nanotubes, from 0 to 3 weight percent. They meticulously evaluated these materials using electrochemical impedance spectroscopy, alongside measurements of hydration capacity. This allowed them to characterize the electrical properties under both liquid and dry conditions. Their primary goal was to identify the optimal formulation for intraoral EEG electrodes. So what did they find? Crucially, the composite material containing 3% carbon nanotubes demonstrated the most suitable electrochemical properties. This formulation showed significantly improved ionic and electronic conductivity compared to the undoped hydrogel. This enhancement was attributed to better CNT dispersion and polymer cross-linking within the material. These findings strongly support the potential of this specific composite for intraoral EEG applications. In conclusion, this research is a significant step forward in wearable neurotechnology. The identification of the 3% CNT hydrogel composite as an optimal material provides a strong foundation for developing next-generation 3D printable intraoral EEG electrodes. This work not only advances our understanding of CNT hydrogel properties, but also paves the way for future in vitro and in vivo validation of these innovative sensors. We're excited to see where this research leads. Up next, our fifth paper out of 10, CSNK2B gene replacement rescues autism-related phenotypes and establishes translational EEG biomarkers by Chaodong Ding and Zilong Qiu from Department of Neurology, Songjiang Research Institute, Shanghai Key Laboratory of Emotions and Affective Disorders, Songjiang Hospital affiliated to Shanghai Jiao Tong University School of Medicine, Shanghai, China. This paper was selected because it demonstrates a successful gene replacement therapy in a relevant animal model for a specific genetic neurodevelopmental disorder, offering a direct therapeutic strategy and identifying crucial translational biomarkers, which represents a significant. Let's break down its key contributions. Variants in the CSNK2B gene are known to cause severe neurodevelopmental disorders, including autism and epilepsy. Unfortunately, current therapeutic options are limited, and we lack reliable biomarkers to track disease progression or treatment efficacy. This research aims to address these critical gaps by exploring the impact of CSNK2B deficiency and testing a potential gene replacement strategy. Researchers developed CSNK2B haploinsufficient mice to model the human condition, observing key disease features like social deficits and seizures. They then applied an early postnatal, brain-wide gene replacement technique to these mice. The study aimed to understand if this intervention could reverse the observed neurological and behavioral impairments. This approach allowed for a direct investigation into the effects of gene dosage correction. The gene replacement therapy significantly improved survival in the treated mice. Crucially, it also rescued a wide range of disease phenotypes, including structural abnormalities, behavioral deficits, and spontaneous seizures. Furthermore, the treatment normalized key brain activity signatures, such as theta and gamma power, which are vital for circuit function. These findings demonstrate the profound impact of correcting CSNK2B dosage. This study provides compelling evidence that CSNK2B gene replacement can reverse severe neurodevelopmental issues, even when administered postnatally. The identification of translational EEG biomarkers is a major step forward, offering a non-invasive way to track treatment response in future studies. While promising, the next critical step involves translating these findings from mice to human clinical applications, carefully considering safety and efficacy. Up next, our sixth paper out of 10, Loss of SMARCAD1 Mitigates Tauopathy by Vaishnavi S. Jadhav and Brian C. Kramer from Division of Gerontology and Geriatric Medicine, Department of Medicine, University of Washington, Seattle, Washington, USA. This paper was selected because it identifies a novel genetic target, SMARCAD1, that significantly mitigates tauopathy, offering a crucial new avenue for therapeutic development in devastating neurodegenerative diseases like Alzheimer's. Let's break down its key contributions. Tauopathies, like Alzheimer's disease, are devastating neurodegenerative conditions marked by the buildup of misfolded tau protein. Currently, effective treatments are limited, highlighting an urgent need for novel therapeutic strategies. This research aimed to explore new molecular targets that could potentially mitigate the progression of these debilitating diseases. Our study employed a multifaceted approach, primarily utilizing C. elegans transgenic models to investigate genetic modifiers of tauopathy. We combined forward and reverse genetic screens with molecular techniques to identify key pathways. The findings were further validated in mammalian cell lines and contextualized by examining human post-mortem AD brain tissue. We found that losing SMARCAD1 function dramatically rescued both neuronal dysfunction and neurodegeneration in our C. elegans models. This beneficial effect was linked to a significant decrease in both phosphorylated and total tau protein levels, which we traced back to a reduction in tau mRNA transcripts. Furthermore, we observed that SMARCAD1 loss corrected abnormal H3K9Me3 chromatin methylation, suggesting a novel epigenetic mechanism. This study highlights SMARCAD1 as a compelling new target for developing therapies against tauopathies, offering a novel mechanism to reduce tau protein. While our findings are robust across C. elegans and cell lines, further in vivo validation in more complex mammalian models is crucial. Moving forward, investigating specific SMARCAD1 inhibitors could pave the way for innovative treatments for diseases like Alzheimer's. Up next, our 7th paper out of 10, a molecular protein fusion framework for rapid virtual screening, accelerating lead discovery for undruggable, oncogenic targets by Qinxi Zhou and Dengming Ming from College of Biotechnology and Pharmaceutical Engineering, Nanjing Tech University, 30 South Puzhu Road, Jiangbei New District, Nanjing, China. This paper introduces a highly novel machine learning framework, MPFFIS, that integrates protein language models and graph neural networks to accelerate virtual screening for notoriously undruggable oncogenic targets like. Let's break down its key contributions. Today, we're diving into a critical challenge in cancer research, targeting KRAS G12D, a notoriously difficult oncogenic mutation found in pancreatic cancer. This mutation has long been considered undruggable due to its limited binding pockets, making it incredibly hard to develop effective therapies. Our research aimed to tackle this by developing a novel computational framework. We wanted to see if we could accelerate the discovery of new inhibitors for this challenging target. Our study developed a novel computational framework called MPFFIS. This framework integrated advanced machine learning models, specifically the ESM2 protein language model and an MPNNGNN molecular graph network, to jointly learn features from both proteins and compounds. We trained this model on existing KRAS G12D inhibitor data and then used it to virtually screen over 134,000 molecules. Subsequent validation involved detailed Autodoc Vena docking and GROMACS molecular dynamics simulations to confirm binding and stability. The MPFFIS framework achieved favorable predictive performance on our test dataset. Most excitingly, it identified a remarkable 2663 candidate compounds from over 134,000 screen molecules. Several of these candidates showed favorable binding affinity and enhanced structural stability when compared to known reference inhibitors like MRTX 1133. Our molecular dynamics simulations further supported the stability of these predicted complexes, confirming the involvement of key binding residues within the KRAS G12D pocket. This study demonstrates that our MPFFIS framework can efficiently identify potential inhibitors for challenging targets like KRAS G12D, offering a powerful new tool for drug discovery. While our computational results are highly promising, the next crucial step involves rigorous experimental validation to confirm the efficacy of these candidate compounds in the lab and in living systems. Ultimately, this framework has the potential to be adapted for other undruggable targets, significantly accelerating the development of precision medicines across a range of diseases. Up next, our 8th paper out of 10, high-fidelity genome and prime editing enabled by the AI-designed OpenCRISPR, one by Haiyun Huang and Daesik Kim from Department of Precision Medicine, Sungkyunkwan University School of Medicine, Suwon, Republic of Korea. This paper was selected because it introduces an AI-designed, high-fidelity CRISPR system, OpenCRISPR, one, that significantly advances genome and prime editing by improving target specificity without compromising on. Let's break down its key contributions. CRISPR, Cas9 technology has truly transformed our ability to edit genomes, opening doors to countless research and therapeutic possibilities. However, a persistent challenge has been balancing the system's on-target editing efficiency with its specificity, meaning avoiding unintended edits elsewhere in the genome. Often, improving one comes at the cost of the other. This paper addresses that fundamental limitation, seeking a solution that offers the best of both worlds. Researchers conducted head-to-head comparisons in various human cell lines, including HEK 293T, iPSCs, and MRC 5 fibroblasts. They evaluated the novel AI-designed OpenCRISPR, one nuclease against standard Cas9 and high-fidelity Cas9 variants, as well as its prime editor versions against existing prime editors. Key outcomes measured included on-target editing efficiency, the extent of off-target mutations using advanced sequencing methods, and the overall specificity ratio. The system was even tested for delivery using engineered virus-like particles, mimicking a clinically relevant approach. The results were quite striking. OpenCRISPR-1 demonstrated editing efficiency comparable to standard Cas9 across multiple genomic loci. Crucially, it achieved up to a 553-fold reduction in off-target mutations compared to Cas9, and its off-target indices matched or even surpassed those of current high-fidelity Cas9 variants. Furthermore, when converted into a prime editor, OpenCRISPR-1 lowered the relative specificity ratio by an impressive 97%, indicating superior precision in more complex editing tasks. These numbers clearly show a significant leap forward in genome editing precision. This research is truly groundbreaking, as OpenCRISPR-1 represents a significant advancement in genome editing, finally overcoming the critical specificity-efficiency trade-off. It showcases the immense potential of generative AI in designing novel proteins with tailored properties, opening a new era for rational protein design. Looking ahead, the next crucial steps involve evaluating OpenCRISPR-1's performance in living organisms and exploring its direct therapeutic applications for various genetic diseases. This technology holds great promise for both research and clinical settings. Up next, our 9th paper out of 10, integrating phenotypic and genomic data with machine learning to predict antimicrobial resistance and identify genetic biomarkers in E. coli by Sarah Halalia Adeyemi and Roshan Patel from Bioinformatics Program, Department of Computer Science, School of Computer Science, Mathematics and Natural Sciences, Morgan State University, Baltimore, Maryland, USA. This paper was selected because it introduces a highly impactful machine learning framework that integrates phenotypic and genomic data to predict antimicrobial resistance in E. coli, directly addressing one of the most. Let's break down its key contributions. Antimicrobial resistance, especially in bacteria like E. coli, poses a significant global health threat. We're seeing increasing resistance to common antibiotics, making infections harder to treat. This research aimed to tackle this challenge by exploring if machine learning could help us better understand and predict this resistance, ultimately improving patient care. Our study utilized a large dataset of over 17,000 E. coli clinical isolates from the BVBRC. We employed various machine learning models, including XGBoost and Random Forest, to predict antimicrobial resistance. The approach involved integrating antimicrobial susceptibility testing data with genomic information to identify key resistance determinants. We also performed phylogenetic analysis to understand resistance patterns. We found that the XGBoost model was the top performer, achieving an impressive 0.86 accuracy and a 0.932 ROC/AUC in predicting resistance. The Random Forest model also showed strong results with 0.82 accuracy. Crucially, our analysis pinpointed several genomic biomarkers, such as GIRA and CTX-M15, which are key drivers of this resistance. These findings highlight the power of integrating diverse data types. This study demonstrates a powerful new approach for predicting antimicrobial resistance, combining machine learning with genomic insights. Identifying specific genetic biomarkers can pave the way for better diagnostics and targeted interventions. While promising, future research should focus on validating these models in real-world clinical settings and exploring their real-time application to truly impact patient care. Up next, our 10th paper out of 10, Prenatal Alcohol Exposure Predicts Academic Outcomes from Childhood to Adolescence. A prospective longitudinal study based on meconium ethyl glucuronide by Stefan Mesterman and the IMAC-MIND Consortium from Department of Child and Adolescent Mental Health, University Hospital Erlangen, Friedrich Alexander Universität Erlangen-Nürnberg, FEU. Erlangen, Germany. This paper was selected because it provides robust, longitudinal evidence linking objectively measured prenatal alcohol exposure to long-term academic outcomes, addressing a critical public health problem with significant implications for intervention and policy. Let's break down its key contributions. Good morning, everyone. Today, we're diving into a crucial topic, the long-term effects of prenatal alcohol exposure, or PE, on a child's academic journey. We know PE can cause developmental issues, but understanding its real-world impact on school performance is vital for early intervention and support. This study aimed to shed light on this important gap in our knowledge. Researchers conducted a prospective longitudinal study with 156 participants. They assessed prenatal alcohol exposure using two methods, meconium ETG and maternal self-report during the third trimester. They then followed these children, comparing their academic performance at primary school age, around 6 to 10 years, and later at secondary school age, between 12 and 14 years, to see how PE influenced their educational paths. The findings were quite telling. At primary school age, children with maternal self-reported PE showed significantly lower school grades, while ETG-positive children also showed a trend towards lower grades. Moving into adolescence, both groups with PE had an almost twofold increased odds of being placed in lower educational tracks. These results clearly indicate a persistent negative impact of PE on academic achievement. This study underscores the profound and lasting impact of prenatal alcohol exposure on a child's academic journey, from primary school right through to adolescence. It highlights that academic performance is a critical indicator of neurodevelopmental challenges stemming from PE. Importantly, the use of meconium ETG could be a game changer, allowing us to identify affected children earlier and provide timely interventions to support their educational success. This research truly emphasizes the importance of prevention and early detection. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.